Hello, hi guys. I hope you can see my screen. I am not at my home right now. I am at some place else. So there'll be maybe some visual and connectivity problems. However, I'll try my best, to be quick and at least upload the first video of 2025 in the series that we are going on about right now. So in the last part of CML and its molecular monitoring, we discussed the various pathogenic pathways that can lead to formation of a chronic myelogenous leukemia. We all know about the tyrosine kinase pathway, but apart from activation of the tyrosine kinase enzyme, the pathways, the map, etc., pathways leading to that, there is a role of epigenetics, there is role of IKZF4 and other pathways which can also cause a CML to occur in a patient. Now, since that part was covered, the next today's part is of very short 8 slides where I'll be touching upon the diagnostic modalities that are utilized commonly in diagnosing CML. You have read about, I took what is CML, what are the WHO criteria for CML, what is the basic pathogenesis of CML. You can go ahead and read, listen to all these short lectures together. Today, we all know that to diagnose CML, once you've gone through the first few steps, if you come to the molecular diagnosis of CML, you have to show the presence of BCR-ABL translocation, document its presence, and that is the most important diagnostic modality. So the image which is flashing in front of you is that of fluorescence in situ hybridization, or FISH. One of the modalities, this is one of the very common modalities, very cost effective modalities where you are going for a molecular technique which is used to diagnose CML, where this is the basic color coding of a FISH kit, where there are two red, two green signals, or one red or one green signal, and then you get a fusion signal. So normally, in a normal case, if I just stick to I'm not going to tell you in detail about what fish is. I've already done that once. What I'm just going to tell you is, when you apply fish on a case of CML, what would you get on the slide? This is done on a fluorescent microscope. So if I take it is a fusion probe over here. If you can see, the yellow, this you must remember from my previous lectures, that when we are applying fish, it can be a break apart probe that we are reading, a fusion probe that we are reading, depends, you should know what you want to see. So a fusion probe normally is of yellow color. So fish is based on the concept that you know which mutation you have to study. So I know that I have to see the translocation and the fusion product between 9 and 22. So I will use a probe which normally stains the 9 and the 22 in a chromosome. So if I take a normal cell, put this stain on that cell, on the chromosome of that cell, after all the processing that takes place in fish, in a normal person, since there are two set of chromosomes, I should get two green and two red signals, which is normal. Two red signals are the ones which are painting chromosome 9, two green signals from chromosome number 22. But when the 9 and 22 translocate, now this probe is made in a way that it attaches to that end which normally breaks, to that part of chromosome which breaks and translocates. So here there is no translocation, so the 22 sticks to 22 and the dye which is labeling 9 sticks to 9. Now this is not a simple protein reaction. Remember that you are reading a chromosome. This blue is a nucleus. Inside that there are chromosomes. Remember that. So you are reading a chromosome. So actually reading amino acid pattern. So the probe that is labeled in my kit is an amino acid pattern. Don't forget that. Now, when a breakage, supposing this part translocates here and fuses, so now the color that should come when a translocation 922 occurs is yellow. So if I get yellow signals, because there is double translocation, so if I'm getting yellow signals in a slide where only red or green should be seen, that means there is BCR-ABL positivity. Easy to interpret. So fish is the most commonly used technique to document BCR-ABL. But this was not enough. We also need to not only know the presence of BCR-ABL, that is qualitative analysis, we also need to do the quantitative analysis. Hence came the role of RT-PCR. RT-PCR can be done in two ways. One, it only documents whether the mutation product is present or not, and second is how much quantity of that mutation product is present, is also known as RQ-PCR or quantitative PCR. Okay, both have separate principles. I'm not going to go into the details of the principles per se, but I'll show you what they are. So this is karyotyping, where you see where that part of 9 and 22 gets translocated, one gets bigger, one gets smaller. So I've already done this. Then RT-PCR and RQ-PCR. Karyotyping, just for you to know, in European countries or outside, yes, but in India, we do not rely too much on karyotyping where CML is concerned. That is why I'm not going into too much detail. You might get cases where a myeloproliferative neoplasm progressed into an AML, and there they did a karyotyping. There it becomes important even in India. And they saw this 922 translocation with other translocations. Right? But in India, we normally just stick to PCR, or at max fish, we normally don't do karyotyping in a plain, simple, straightforward case of CML. Okay. So again, this is the algorithm which says, you have blood, you extract RNA, then you create a complementary DNA, and then you do both these types of PCRs. So I had shown this image when I had started, that what are the complementary regions which get translocated, and based on what part of 9 and 22 are getting translocated, you get these byproducts P210, P190 or P210 variant. This P210 variant is cryptic BCR-ABL translocation. So if I do a PCR and I use probes which are only for P210 or P190 but do not contain cryptic, and mostly this happens, we can't produce a mixed bag of probes for every known BCR-ABL mutation. We do it for common mutation, so that when the probe goes and sticks to that end, a flash of fluorescence or a flash is seen which is captured in Sanger sequence. So we normally can't do it for everything. So sometimes you get a clinical picture of MPN, you get the patient responding to the treatment, but you are not getting a documentation of a BCR-ABL. Earlier days, this was known as atypical CML. Now they are changing the names and the terminologies, but in earlier days, those CMLs where you could not express a BCR-ABL, they were behaving like an MPN, they were responding. So those are those cryptic breakpoints in that BCR-ABL region. Okay. So this is another, when you are do when we when we put our products in this thermal cycler and they get amplified. So capillary electrophoresis is a way where we can see, like supposing this is we have a standard and a ladder is run, where we have test which is being run, controls which are being run, and then we have a standard. Standard is, supposing I want to see BCR-ABL. So my positive control, my positive control is, if I want to see a BCR-ABL on the capillary electrophoresis machine, there's a complete, this kind of a band graph or a histogram comes on the computer screen. So I know that at this location, if any band comes in a test sample, so a test sample can be run over here, over here. So and this is your positive control, and similarly there'll be a negative control. So if I'm getting a band in this location, in a case this band is known, I know that the probe kit that I'm using is for BCR-ABL fusion product. So if in my sample I get a band in this location, then I know that this sample has BCR-ABL mutation. So this is a qualitative analysis. Then I put this whole thing, I put it in my light cycler and get a quantitative analysis, see how much BCR-ABL is present. That is important because you have to assess the molecular response, whether a morphological response, cytogenetic response, then molecular, major molecular, deep molecular response. Log reduction of amount of BCR-ABL is calculated to see कि कितना copy number कम हो रहा है, 3 months, 6 months, 12 months. We go on doing it till it becomes, I'll show you what it is. Deep molecular response, major molecular response, what it is, I'll show you, right. So this is how you calculate the quantitative. I'm not going in detail, it's not important for you. So quantification of BCR. So when we quantify BCR-ABL, when we always see when you are finding the amount of something in a test sample, you should also have a control and find the amount of something in a control sample so that you know that your test is being run correctly. Every time, even if you're running a CBC, you first do a quality control check. So we take ABL1, our kit has ABL1 as the control gene. We will measure the amount of AVL1 in our control samples, which we are running parallel to the test samples that we are having, where we are reading the BCR ABL1 mutated product. And then we see a ratio of both of these. So how do you quality check? The ABL1 should be more than or greater than 10,000 molecules per sample. The copy, the final product of when putting it through a light cycler, we should get more than 10,000 ABL1 molecules. Then only can we say, okay, now we can run our test sample. It will be able to quantify the amount of BCR-ABL mutated product correctly. Okay.